pattern separation and mood regulation, and its alterations in aging and neurodegenerative diseases. Adult hippocampal neurogenesis is a dynamic process that encompasses various maturation stages, in which immature neurons express distinctive markers specific of each stage. Throughout their maturation, these newborn neurons acquire characteristic morphology, since they integrate into neural circuits. And when they integrate in these neural circuits, they increase the plasticity, the structural plasticity of the dentate gyrus. Adult hippocampal neurogenesis has been widely studied in rodents and non-human primates since its discovery in 1963. In 1998, Erickson suggested the occurrence of this process in humans. A cohort of patients with cancer were treated with VRDU, and after they died, Erickson observed VRDU-positive dentate granule cells. Subsequently, human adult hippocampal neurogenesis has been studied by using multiple techniques, such as the incorporation of carbon-14, Western blot, RNA-scope, and immunohistochemistry. And our laboratory is mainly focused in the immunohistochemistry protocol, and we have worked to develop a robust methodology to study adult hippocampal neurogenesis in post-mortem human samples. I would like to summarize a bit this process, and firstly, we dissected the hippocampus of the brain, and this block of the hippocampus is fixed for 24 hours in 4% paraformaldehyde. As is widely known, prolonged fixation abolishes the visualization of some antibody signal, including most of adult hippocampal neurogenesis markers. Next, we included this block in sucrose agarose, and we dissectioned it in a vibratome. We determined that the samples needed to be pretreated with sodium borohydride and heat-mediated citrate buffer antigen retrieval in order to eliminate the autofluorescence caused by aldehyde fixation and to visualize adult hippocampal neurogenesis. Next, most of antibodies related with the adult hippocampal neurogenesis process are visualized using a standard immunohistochemistry buffer. However, in our last work, we determined that some more sensitive epitopes, which is the case of nestin, needed a milder detergent, which is the saponin. So, using this change in the immunohistochemistry protocol, we were able to visualize neural stem cells in the human dentate gyrus. Next, the autofluorescence should be further eliminated before the samples are observed under the confocal microscopy. So once we have set up the protocol to study adult hippocampal neurogenesis in humans, we wonder whether this process was similar to that in rodents. So with the help of Dr. Alberto Rábano, we studied a cohort of 15 neurologically healthy control subjects. Firstly, we focus on determining the presence of radial glia-like cells in the human dentate gyrus. So because these cells are thought to be the source of new neurons, and their existence had been debated in the last decades. So using this modification in the protocol that I have previously said, we were able to observe multiple neuronal markers validated in rodents, such as GFAP, Nextin, SOX2, and Vimentin. And these cells are predominantly located in the subgranular zone. Therefore, to further characterize the neural stem cell phenotype of these cells, and since some of these markers could be also expressed by non-astrocytic cells, we studied the S100 beta marker, that is an astrocytic marker, in nestin-positive cells. And we studied the morphology and their location. Nestin-positive S100 beta negative cells have a longer process, and they are predominantly located in the subgranular zone. Moreover, nestin-positive cells do not show correlative variation with S100 beta positive astrocytes. So this indicates that nestin-positive S100 beta negative cells are not astrocytes, but radial glia-like cells with a neural stem cell phenotype. Neural stem cells asymmetrically divide and give rise to proliferative cells. We use some markers to detect these proliferative cells, phosphohistone 3, that is a mitosis marker, and HUC-HUD, that is a marker of proliferative neuroblast. We observed that these cells were predominantly located in the subgranular zone. Moreover, proliferative neuroblasts, 90% of these proliferative neuroblasts were also positive for the immature neuronal marker double cortin. So this indicates that in the human dentate gyrus, most of the proliferative cells are transit-amplifying progenitors and proliferative neuroblasts. And also, these data are in agreement with previous that indicate 700 new neurons being incorporated in the human dentate gyrus daily. So with all these results, we can say that the presence of neural stem cells and proliferative cells supports the idea that the pool of neural stem cells is preserved throughout life. Next, we study the immature newborn neurons, and double cortin is a microtubule-associated protein and is the gold standard marker of adult hippocampal neurogenesis. This marker is, and double cortin also is expressed since neuroblast and to the last immaturity stages of the process. And double cortin is not expressed by astrocytes, is not expressed by microglia, is not expressed by the vasculature structure, and is not expressed by the interneurons. Conversely, double cortin positive cells also express PROS1 and other markers of the maturation, such as neuroblast and markers of the initial and of the last stages of immaturity. Double cortin positive cells colocalize with markers of distinct markers of other maturative stages. In the initial stages, we can colocalize double cortin with calretinin. The immaturity stages, we can use the NUN marker, and in the last stages, we can colocalize double cortin with calbindin, that is a marker of mature neurons. So with this, we can study the development of immature dentate granule cells, and we can determine with the morphology and the position the levels of maturation of these immature neurons. So we studied the area of the soma, the number and the orientation of the neurites, and also the position. So in the initial stages of maturation, the cells have a smaller soma and multiple horizontal neurites, and they are mainly localized in the subgranular zone. In the intermediate stages, the cells have bigger somas, and they start to have one apical neurite, and they are localized in the granular cell layer. And in the last stages of maturations, the newborn neurons have bigger soma and have one apical, one vertical dendrite through which they expand the dendritic tree, and they are mainly localized in the granular cell layer. So with all this data, we can say that adult hippocampal neurogenesis is a dynamic and robust process that exists until the ninth decade of human life. So in rodents, this process of adult hippocampal neurogenesis is supported by the neurogenic niche. The main components of the neurogenic niche are the blood vessels, the astrocytes, and the microglia. In humans, we have also observed this type of cells that may putatively be orchestrating adult hippocampal neurogenesis throughout aging. So we have observed a profuse red of capillaries that occupy 7% of the dentate gyrus. Moreover, we have seen astrocytes and microglial cells. So we can say that human and rodent neurogenic niche share similarities. Microglial cells have specialist membranous structures named the phagocytic pouches, which are responsible of engulf dysfunctional cells that are shown here as pycnotic cells. Moreover, microglial cell morphology correlate with the presence of phagocytic pouches. So, microglial cells with no pouches present long non-polarized process, while microglia with 1, 2, or more than 2 pouches have a profusely branched process and polarized morphology. Moreover, the number of phagocytic pouches decrease with age, pointing to a dysfunctional microglial phenotype in aged neurologically healthy control subjects. So regarding all this data, we and others laboratories have shown a reduction in adult hippocampal neurogenesis during physiological aging, and moreover, adult hippocampal neurogenesis is altered at multiple levels in Alzheimer's disease. So we wonder whether this process was also altered in other neurodegenerative diseases, such as amyotrophic lateral sclerosis, Huntington disease, Parkinson's disease, dementia with Lewy body, or frontotemporal dementia. Importantly, the hippocampus is not the main brain area affected in this patient, but a variable degree of hippocampal alteration could be observed in some of these patients. So the first neurodegenerative disease that we studied was ALS, that is characterized by the degeneration of motor neurons, which causes muscular atrophy. Some of these patients present memory impairments and which could be related to hippocampal dysfunctionality. So we wanted to study how was the process of adult hippocampal neurogenesis and the neurogenic niche in these patients. We observed that the number of proliferative cells didn't change, but there was an increase in neural stem cells and in mature dentate cranial cells. This should not be interpreted as an increase in adult hippocampal neurogenesis, due to the morphology of these neurons is altered in the last stages. Moreover, we observe an increase of pycnotic cells and also alterations in the neurogenic niche, as we can observe here with an increase of astrogliosis, a decrease in the number of phagocytic pouches of the microglial cells, and an increase in the increase of the dentate gyrus capillary thickness. So to sum up these slides, ALS patients present an increase in neural stem cells and in mature dentate cranial cells, and the morphology of the immature neurons is altered, and the numbers of astrocytes and the pycnotic cells are increased, and the microglial cells are altered. So all these results indicate that both adult hippocampal neurogenesis and the neurogenic niche are vulnerable to ALS. The next neurodegenerative disease that we studied was Huntington's disease. It's a genetic disorder caused by the repetition of CAG triplets of Huntington, which causes the death of striatal interneurons. Some of these patients present moved and cognitive comorbidities, which also could be related to the hippocampus. So we wanted to know how was the adult hippocampal neurogenesis and the neurogenic niche in these patients. We observed an increase in neural stem cells and in immature dentate cranial cells, whereas the number of proliferative cells remained unchanged. The morphology of these cells, of these immature neuroneurons, were altered, as we can observe in the initial stages, and also we can observe a decrease in NEN-positive cells, and the maturation of these cells is altered in the initial stages. Moreover, as we have observed in ALS, we can see an increase in pycnotic cells and also alteration in the neurogenic niche. Here you can see an increase in S100 beta-positive astrocytes and a decrease in the phagocytic index of microglia, and also an increase in the dentate gyrus capillary thickness. So just to sum up these slides, Huntington patients present an increase in neural stem cells, in immature dentate cranial cells, and in the total dentate cranial cells. And also, the immature neurons have the morphology and the differentiation altered. And respect to the neurogenic niche, we can observe an increase in astrocytes, in the vasculature, and in the apoptosis. And also, the morphology of the microglia is altered. So, with all these results, we can say that adult hippocampal neurogenesis and the neurogenic niche are vulnerable to Huntington disease. The next family of disorders that we studied were alpha-synucleinopathies that are characterized by the accumulation of alpha-synuclein aggregates in the brain. Two of the disorders were dementia with Lewy body and Parkinson's disease, which have diverse clinical manifestation, including cognitive and motor impairments. In this slide, data obtained from patients with dementia with Lewy body are represented in blue, and the data from patients with Parkinson's disease represented in dark blue. So in these patients, we observe an increase in neural stem cells, and particularly in Parkinson, there is an increase in proliferative cells and in immature dentate cranial cells, and the morphology of the latter cells is altered in both alpha-synucleinopathies. And as we have observed in ALS patients and Huntington disease patients, there is also an increment of the numbers of pycnotic cells in these patients that are accompanied with alterations in the neurogenic niche, such as alterations in the microglial cells and in the dentate gyrus capillary thickness. And moreover, there is an increase of astrocytes in Parkinson's disease. So to summarize this slide, we observe that patients with dementia with Lewy body present arterial morphology of the immature neurons, whereas patients with Parkinson's disease present an increase in neural stem cells, a proliferation, and immature dentate cranial cells, and the morphology and differentiation of the immature neurons is also altered. In patients with dementia with Lewy body, there is an increase in the vasculature and apoptosis, and the microglia is altered, whereas in patients with Parkinson's disease, we can observe an increase in astrocytes, in apoptosis, and also an alteration in the microglial cells. So we observe that in adult hippocampal neurogenesis and the neurogenic niche present higher vulnerability to Parkinson's disease than to dementia with Lewy body. And the last neurodegenerative disease that we studied was frontotemporal dementia. That is one of the most prevalent forms of early onset dementia. These patients present temporal lobe atrophy and might develop cognitive and motor impairment. So we wonder whether this process were also altered in these patients. We observed that there were no change in the neural stem cells, while the proliferation is decreased in these patients. All the number of immature neurons weren't also changed, but there is an alteration in the morphology of these cells. As we have observed in the rest of the neurodegenerative diseases, in this one we also observe an increase in the pyknotic cells, and the neurogenic niche is also altered, as we observe an increase in astrocytes, in the dentate gyrus capillary thickness, and a decrease in the phagocytic index of the microglial cells. So to sum up these slides, patients with frontotemporal dementia present a decrease in the proliferation, and the morphology and differentiation of the immature neurons is altered. And we also observe an increase in the astrocytes, in the vasculature, and in the apoptosis. So adult hippocampal neurogenesis and the neurogenic niche present a mild vulnerability to frontotemporal dementia. So, in conclusion, our data support the persistence of neural stem cells, proliferative cells, a dynamic population of immature neurons, and the neurogenic niche in the human dentate gyrus throughout life. Moreover, our data support the differential vulnerability of human adult hippocampal neurogenesis to distinct neurodegenerative diseases. The heterogeneous nature of the hippocampus alteration present in these patients likely underlie the existence of a specific disease-associated adult hippocampal neurogenesis signatures. In patients with ALS, we can see a late blockade of immature neuron maturation, while in patients with Huntington's disease, that retention is observed in the initial stages. In patients with Parkinson's disease, we can observe more severe alterations in adult hippocampal neurogenesis and the neurogenic niche than patients with dementia with Lewy body. And in patients with frontotemporal dementia, we can observe a mild alteration in adult hippocampal neurogenesis and the neurogenic niche. So, our data suggests adult hippocampal neurogenesis impairment as a relevant mechanism underlying hippocampal dysfunction in age and disease subjects. So, finally, I would like to thank the wonderful members of my lab for their support and Maria for her guidance, and without them this work would have not been possible. And moreover, I would like to thank the former members of the lab, our collaboration, and all the organizations that have financed this project. And finally, I would like to thank you all, and I will be happy to take questions.
Thank you very much, Julia. Very exciting stuff. Paper is open for discussions if there are one or two questions in the audience.
Congratulations, Julia. Excellent work. A question about FTD, frontotemporal dementia. Is tau dependent or is TDP-43 dependent, the sample that you have? What about frontotemporal dementia, if it is tau dependent or is TDP-43 dependent, this kind of dementia?
In these patients, we have phospho-tau and also TDP-43. We have both alterations in the dentate gyrus. We can observe the accumulation of both aggregates.
There are no difference between both in the data that you have presented?
We haven't checked that.
Okay.
We haven't checked that. Thank you.
Yep.
Congratulations also, Julia. I was wondering, because you have compared so many neurodegenerative diseases, I was wondering how you guys handled the age effect I mean, were they matched by age? All the samples? How did you approach the problem of the aging?
Yes, all our samples from the neurodegenerative diseases have age-matched controls. We have controls to compare each disease.
And can you see the progression then along the timeline?
In most of the markers, we don't observe a difference with the age. Only in control subjects, we observe the decrease in double cortin positive cell that was previously described. But in most of the markers, we don't observe a difference with aging.
Okay, thank you.
Okay, thank you. Okay.
Do you see changes in vessel thickness? What do you think is this? What is your interpretation of these changes in vessel thickness?
Okay, yes, we observe in most of the disease, we observe this increase in vessel thickness, and we think that it could be because of the distal alterations and also because of the hippocampal alteration. And we don't know if these are neuroprotect mechanisms or if it's a neurodegeneration mechanism, because in the blood vessels, all the substances are going to the dentate gyrus, so we don't know if it's a neuroprotective effect or a neurodegenerative disease effect. Sorry.
Another one. Have you considered the possibility of checking the aggregates of these different diseases in these samples to see whether they are closer or farther away from the niche?
Yeah,
Aggregates in the niche in these diseases. Have you looked at that?
We have seen the aggregates in the dentate gyrus, and we also try to correlate this aggregate with some of our markers, and in most of the pathologies we didn't observe a correlation between them, but in some of them that are specific ones in the paper are better described, we observed some correlation between them, but we haven't checked if they are more near of the cells in the neurogenic niche or the localization. We just saw that they are in the dentate gyrus.
All right, thank you very much. So and now I'd like to call to the podium, our scientific director, Adolfo López de Muniain, and the coordinator of the training program, Teresa Iglesias, to present the award to Julia.